Thank you all for coming. I'm KWilson, and part of my job in the department is actually outreach into schools, so it's part of my job description. When this new molecular biology for level 3 came in, probably in the mid-90s, I ran quite a few one-day courses for teachers just to update them on some of the content. And then we realised there was actually quite a need for kids to be able to come and do some prac work, because schools are quite limited in the amount of equipment they've got and what they're allowed to actually use in schools. And so what I do is, I bring year 30 students come into the department. We've decided to make it for 3 hours, and that makes it manageable, so they're not encroaching too much on the rest of their subjects. Some of the other teachers get a bit titchy when they take these students up too long, so they come in for either a morning or an afternoon. It's free of charge, so we don't actually charge the school. The only problem is we can only have them in June and October, so it's only when our labs are not being used and the prep room are not so busy. So sometimes that's a little bit of a constraint, but I actually have quite a few schools booked in already for June next year. So it's really popular. And the thing I've realised is it really doesn't take much to actually get the kids excited. So they come in and the first thing they do is put a lab coat on, and then they put gloves on, latex gloves, so I think that's really exciting. Get to use the pipetters for the first time ever, the micro pipetters. And so really if I just sent them away, they'd probably be quite excited just with that. But then what we do is we do a restriction enzyme digest using PA plasmid. So we use two restriction enzymes, EcoR1 and BMH1. They separate the fragments using electrophoresis while the gel's running, so it takes about 45 minutes. They go away and can have a coffee for half an hour, so they explore the campus, and then I give them a talk, and I'll tell you about the talk, but I'll give them in just a minute. So this is a picture of the plasmid that we actually use. So that's the PA plasmid, so there's quite a lot of learning just in that. So you can see it's actually had a piece of human DNA engineered into it, so that's another piece of learning material, and you can see there's an EcoR1 site on each side of that insert, which they're actually going to cut out, and you can see there's some BMH1 sites as well. So there's quite a lot of learning just in that piece. So I do a basic experiment with them, and then the school teachers can go and do some extension activity when they go back to the class. So this is a picture of the gel that they get. Now they do everything themselves. They set up the digest, they pour the gels, they load their gels, so it's in pairs, and so each student has a chance to load 2 wells. So you can see this is the marker lane on the outside, so they can talk about the markers. This is uncut plasmid here, so you can see that there's the 2 fragments which are quite characteristic of them uncut, and so you can talk about supercoiling and things. This is the EcoR1, and this larger fragment here is that piece of human DNA that we talked about, and this is the BMH1s. So you can see there's 2 here and 1 down here. So the students get a copy of that each to take away, and so as I said, the teachers when they get back to school can do further activities. So they can work out the sizes of the markers, and then work out the size of the fragments and things, and they love taking that away. So that's really the prac activity, so that takes 2.5 to 3 hours. And as well as that, Bronwyn, who's working the video, she's designed quite a number of posters. And so while the gel is running, I actually talk about one of the posters, and I'll talk about that in a minute. There's quite a lot of them outside on the wall, so you can have a look at those. So part of the curriculum, they have to actually show how the techniques that they use meet a human need. So they have to, sort of show the relevance, if you like. So what Bronwyn's done is she's made these posters where she uses examples of research from within the department, and they do show how the techniques that they've been using meet a human need. There's quite a lot of supplementary notes for teachers, so this one that I'm going to talk about has got quite a few pages of supplementary notes. And we had sent them to all high schools nationally, but it's like all things, they sort of get filed into somewhere, and how we get the teachers to use them is another problem. So the favourite one that I'm going to talk about today, which I talk about when these students are in, is this is the size of the poster here. It's on blindness, and it's based on research that was done by Dr. Mike Denton, who's now retired. And so the first part of the poster just has a little bit about the anatomy of the eye, so I don't do too much about that. And the gene that we're interested in is this RPE65, and it's, this cartoon just relates the actual gene to the function of the gene, so we can talk a little bit about that. So this brings in what they've probably done in year 12.